Hello students, Assalamu alaikum. This is Laiba Tariq, your biology instructor from tutoria.pk. Today we are discussing the exercise of your chapter Chromosomes and DNA. Let's start with the MCQ. So the first MCQ is chromosomes are made up of proteins plus pectin, RNA plus DNA, DNA plus histones or DNA only. The right option is option C, DNA plus histones. Second है, in his work with pneumonia causing bacteria, the mice Griff found that a heat-kill pathogenic cell caused pneumonia. Some substances from the pathogenic cells were transferred to the non-pathogenic cells, making them pathogenic. The polysaccharide coat of the bacteria caused pneumonia. Bacteriophage injected the DNA into the bacteria. The right option is option B. Some substances from the pathogenic cells were transferred to the non-pathogenic cells. Question 3 है, a biochemist isolate and purify the molecules needed for the DNA replication. When DNA is added, replication occurs, but which DNA molecule consists of a normal strand paired with the numerous segments of the DNA a few hundred nucleotides long, which has probably been left out of the mixture? तो इसमें है right option DNA polymerases, DNA ligases, Okazaki fragment या primase. The right option है DNA polymerases because they are involved in the DNA replication. फिर what is the basis for the differences in how the leading and lagging strands of the DNA molecules are synthesized? Option A, the origin of replication occur at 5' only. Option B, the DNA ligase will only work with 3 to 5' direction. Option C, polymerase can only work on one strand at a time. Option D, DNA polymerase can join a new nucleotide to the 3' end of the growing strand. The right option is option D, because you should know कि जो 5' to 3' एंड के ऊपर जो 3' एंड होगा वो फ्री होगा और उसके ऊपर न्यू न्यूक्लियोटाइड ऐड होते जाएंगे. क्वेश्चन 5 है, सिंथेसिस ऑफ अ न्यू डीएनए स्ट्रैंड यूजुअली बिगन विद आरएनए प्राइमर, डीएनए लाइगेज, डीएनए प्राइमर या ओकाजाकी फ्रैगमेंट. तो राइट ऑप्शन है ऑप्शन ए, आरएनए प्राइमर, बिकॉज यू शुड नो कि डीएनए पॉलीमरेज विदाउट द प्राइमर वर्क नहीं करता. Question 6 है, a submetacentric chromosome has centromere in the center, near an end of the chromosome, slightly away from the center of the chromosome, at the end of the chromosome. The right option is option C, slightly away from the center of the chromosome. Question 7 है, the elongation of the leading strand during the DNA synthesis progress away from the replication fork occur in 3' to 5' direction, produces Okazaki fragments, depend on the action of the DNA polymerases. The right option is option D, depend on the action of the DNA polymerase. Question 8 है, whose pioneer work on the genes used the bacteriophage to show DNA was a part of this experiment which went into the bacterial cell for replication. The right option is option C, Hershey and Chase. Question 9 है, if you want to label amino acid but not the DNA, which of the following radioactive isotope you will use? ऑप्शन ए है 18F, ऑप्शन बी 35S, ऑप्शन सी 14C, ऑप्शन डी 32P. अब यहाँ पे आप देखें कि जो कार्बन होता है वो किसी भी डीएनए या प्रोटीन की सिंथेसिस में नहीं आता. फॉस्फोरस होता है लेकिन फॉस्फोरस न्यूक्लियोटाइड की बॉन्डिंग में होता है. तो राइट ऑप्शन होगा ऑप्शन बी 35S, क्योंकि ये प्रोटीन सिंथेसिस में इन्वॉल्व हो रहा होता है. क्वेश्चन 10 है, इन यूकैरियोटिक सेल ट्रांसक्रिप्शन कैन नॉट बिगन अंटिल the two strands have completely separated and exposure to the promoter. Several transcription factors have bonded to promoter. The DNA introns are removed from the templates. The DNA nucleases have isolated the transcription unit. The right option is option B, several transcription factors. Question 11 है, the anticodon of a particular transfer RNA molecule is complementary to the corresponding triplets. Complementary to the corresponding triplet in ribosomal RNA. Part of the transfer RNA bound to the specific amino acid, catalytic making the transfer RNA. The right option is option A, complementary to the corresponding triplets in the messenger RNA. इसके साथ ही हमारे MCQs कंप्लीट हुए. अब हम डिस्कस कर लेते हैं नेक्स्ट टॉपिक में शॉर्ट क्वेश्चंस ऑफ दिस चैप्टर.